We're very pleased to have Miguel Medina Serpas, PhD, join us. He's coming from the lab of Dr. Todd Briscoe at University of Florida Diabetes Institute, and the title of his talk is a lymphotoxin beta signature in human type 1 diabetes. A little bit about Miguel. He's a graduate research fellow, although newly PhD, I guess, in the lab of Dr. Briscoe at UFDI. He received his BS in biomedical sciences from the University of Central Florida and recently defended his dissertation and received his PhD from the University of Florida. While in the Briscoe lab, Miguel has applied spatial transcriptomics and fluorescent microscopy of the human T1D pancreas and pancreatic lymph nodes to identify cellular phenotypes and gene expression programs that are associated with T1D pathogenesis. And I'm going to be dropping the preprint into the chat, so it's there for everyone. And you can also feel free to drop questions in the chat and he will field them as they go. Thank you so much, Miguel, for joining us. We're really looking forward to this talk. It was a really exciting preprint, and hats off for both you and Greg Golden, who's also on the call. So exciting.
Awesome. Thank you so much for the introduction. And thank you to everybody who's taken time out of their afternoon to, listen to us talk about some of our work in spatial transcriptomics. So without any further ado, I'll go ahead and get started. Okay, so just to briefly start, we know that type 1 diabetes is a chronic autoimmune condition where the insulin-producing beta cell of the islets of Langerhans are attacked by autoimmune-mediated destruction. So insulin is this hormone that's going to help regulate blood glucose levels, and this progressive destruction is ultimately going to result in the symptomatic dysglycemia, thereby making individuals have to require exogenous insulin replacement to help maintain their normal blood glucose levels. We know that the pathology of type 1 diabetes is driven largely by immune dysfunction. We know that this is a characteristic feature of type 1 diabetes. We know if we consider the genetic variants that are associated with type 1, which are highly driven by HLA and a number of other immune modulatory genes, if we take a look at the histopathology, which is going to be largely comprised of these inflammatory lesions known as insulitis, which we know are going to comprise predominantly of immune cells, particularly those of CD8 T cell origin, and also the production of islet antigen-reactive autoantibodies, which also goes ahead and pre-indicates that there is some immune dysfunction and dysfunction of the adaptive immune arm that's contributing to this disease. We also know that type 1 diabetes is an organ-specific autoimmunity. We know that a key tool in investigating the immune dysfunction in type 1 diabetes is investigating immune dysfunction via peripheral blood mononuclear cells. But we also know that the immune phenotypes and the gene expression programs that are identified within PBMCs may not be entirely representative of the immune phenotypes and interactions that are contained within the pancreas microenvironment. So this really does require us to take a look at the disease-relevant tissues, such as the pancreas and also the associated draining pancreatic lymph nodes. So what do we know about the immune dysfunction, and particularly about the histopathology of type 1 diabetes? Well, as I previously mentioned, we know that these inflamed lesions known as insulitis are going to comprise of immune infiltration of the pancreatic islets. And I know that people on this call are very familiar with these lesions. If we take a look at the left, we can see a nice intact islet that's representative of a non-diabetic donor, where we can see a nice organized structure with a co-staining of both glucagon and insulin, with the absence of these brown staining CD3 positive T cells within this organized structure. If we contrast that to an insulitic lesion of a type 1 diabetic islet to the right, we can see that this is a pretty dramatic example of this inflammation, where we can see that there is an abundance of infiltration of these brown staining CD3 positive T cells that are presumably mediating the destruction of these insulin producing beta cells. So we know that the CD8 positive T cell is going to be the most predominant immune subtype within these insulitic lesions, but we also know that the immune presence within these lesions is pretty diverse. And if we take a look at these immunofluorescence stainings, we can see not only the presence of these CD8 positive T cells, but we can also see the representation of B cells, CD4 positive T cells, and also the addition of myeloid cells. So we know that there's a diverse representation of immune cells, yet there's still a little bit more immune phenotyping and a little bit more work to do to understand the gene expression programs that are associated with these islet infiltrating immune cells. Another thing that we know about the pattern of insulitis is that it's quite heterogeneous. For example, if we take a look at this diabetic donor, we can see highlighted within the middle here, we can see these intact islets here. We can see this intact structure with co-staining of both glucagon and insulin with the absence of these brown staining CD3 positive T cells. But within the same lobule and within the same donor, we can contrast that to these islets to the left and to the right respectively, which can show pretty much the full loss of red insulin staining, kind of indicating that these have been depleted of their beta beta cells. And then to the right, we can see kind of this active inflammation where we can see pretty intense infiltration of these CD3 positive T cells within this islet. So we know that insulitis exists within the spectrum of complete, ongoing, and then also kind of fully protected islets within these diabetic pancreas. So there's quite a bit to know. There's quite a bit still to know to ask about how and why this is occurring. So this is where we kind of get to these key knowledge gaps. So really what we are trying to understand here are what are the main differences between affected and unaffected islets? What is driving this protection or the susceptibility to immune infiltration? Second, what is the difference between the immune infiltrates, between those that are actively invading the islets and those that are kind of found within the general exocrine space? And then also, can we understand or can we identify any alterations in cell localization or intercell communication within the pancreas that could kind of provide any further insights to the local inflammation that could be occurring within the tissue? So in order to address these questions, one of the things that we really need is access to these human donor tissues. And through the establishment of various consortia, such as NPOT and HPAP, as I'm sure you guys are familiar with, these consortia have significantly enabled our work, our ability to do this work by providing us access to unique and valuable tissue material from type 1 and at-risk individuals so that we can perform these types of studies. So thanks to these consortia, we've been able to assemble a cohort of donors so that we can investigate these immune phenotypes within the pancreas and the T1D. And to do so, we'll be applying these techniques known as spatial transcriptomics. So for those that are unfamiliar with what spatial transcriptomics are, this is going to refer to a collection of high parameter technologies that enable the detection and measurement of RNA molecules within intact tissue sections. There's a number of different commercially available and fully customizable technologies that are out there, but the main qualities to kind of consider are going to be their ability to resolve a number of transcripts and their cellular resolution. So these can range from multi-cell down to all the way to subcellular resolution, where individual RNA molecules are being detected. They can vary in their transcriptional resolution and depth. So we can look at either a predefined targeted set of genes or taking a look at the whole transcriptome. And then their underlying detection modality, whether transcripts are going to be detected through true quantitation using next generation sequencing, or if we're kind of using a more imaging based approach and trying to measure gene expression based off of the direction of fluorescence. Now, one of the major qualities of spatial transcriptomics that kind of brings this unique quality above other transcriptomic profiling technologies is that this is performed on intact tissue sections, which therefore makes it minimally disruptive. A really nice quality about that is that it helps preserve rare or sensitive cellular states that might otherwise be lost or altered during the tissue dissociation process that's associated with preparing single cell preps for RNA sequencing and other such technologies. So to give a pretty brief overview of the spatial transcriptomic technology that we use for this particular assay, we employ the Visium assay from 10x Genomics. So this is going to be an on-slide next generation sequencing array. It exists in a number of different configurations based off of the number of capture areas and the size of these capture areas, but they all contain the same underlying configuration and underlying capture chemistry. When what they're going to comprise of are these predefined capture areas, which are going to consist of these capture spots. These capture spots are going to be 55 microns in diameter. So an important thing to consider when we discuss the following results is that these 55 micron diameter capture spots are going to be larger than an individual cell. So the gene expression profiles that we detect at each individual spot is going to be representative of the aggregate transcriptome of any cell that's contained within that specific area. So if we take a look at the composition of these individual capture spots, they'll be comprised of these individual oligonucleotide sequences that are covalently bound to the slide. They're going to contain a number of different sequence elements that are important for the underlying chemistry, but what's really important for spatially resolving gene expression is going to be the inclusion of this 16 nucleotide spatial barcode. This is going to be unique within the individual capture spots, but different across them. And this is what's going to allow spatial localization of gene expression. And then the other sequence element that's important to consider is going to be this poly T tail. This is going to be critical for actually hybridizing to the mature mRNA transcript via hybridization of that poly A tail. And through a series of different PCR steps, we can go ahead and create a full sequenceable library from these products. So for our experimental design, what we did was, with help from our collaborators at UPenn and HPAP, we assembled a cohort of 20 donors. This is going to be inclusive of 5 non-diabetic donors, 7 non-diabetic but autoantibody positive donors, and these are going to be individuals with at least one detectable islet reactive autoantibody, and then 8 type 1 diabetic donors, established cases, and these are going to vary in their years of disease duration. And what we were able to do was assemble a collection of FFP embedded pancreas sections. And from that, we performed the spatial transcriptomics. Whenever available, and that happened to be for 16 of our 20 donors, we went ahead and also assayed pancreatic lymph node sections from these individuals. These pancreatic lymph nodes were localized to the same region of isolation of the pancreas section. So, for example, if we assayed a pancreas section that was isolated from the body region, the PLN was also acquired from the body region, for example. So we try to keep at least the regional localization relative to the pancreas the same across both tissues. And I'll let you guys go ahead and read the majority of the findings, as we did a pretty thorough characterization across both tissues. So what I really wanted to highlight for today was going to be some of our key findings that we identified within the pancreatic lymph nodes from this particular cohort. So in looking at the pancreatic lymph nodes, the first thing that we wanted to do was characterize the distinct functional compartments of the human PLN. So using a combination of the histology that so we took a look at H&E stained tissue sections, these exact sections that were processed for Visium spatial, and we took a look at the expression of canonical marker genes that were associated with cell types that are known to be contained within distinct regions of the PLN. And using the combination of those two features, we were able to annotate these into distinct regions, which we have defined here. So you can see that was our T cell zone, our lymphoid follicle, the stromal medulla, and kind of this indistinct region based off of gene expression and histology. When we went ahead and performed unbiased marker gene identification for these individual regions, you can see that we have the expected enrichment of the constituent cell types that are confined within these particular regions. Probably the most identifiable and familiar to most in this audience might be, for example, if we take a look at the T cell zone, we can see this is enriched with a number of canonical T cell genes. Likewise, within the B cell follicles, we can see that this is enriched with a number of B cell associated transcripts, so on and so forth. So once we felt confident in the compartmentalization and regional annotations that we have for these individual tissues, again, because we're looking at these multi-cell resolution capture spots, rather than separating this out into distinct cell types like you would with single cell RNA sequencing, we went ahead and did differential gene expression analysis at the regional or compartment level within these tissues. So we went ahead and did differential expression within these individual compartments across the disease groups, and you can see the individual pairwise comparisons listed below and within these group Venn diagrams. So we just kind of assessed kind of globally the number of differentially expressed genes that were detectable across these different compartments. And if we take a look at the bar graph to the left, we can see that the compartment that was associated with the greatest number of differentially expressed genes was the lymphoid follicle, followed by the medulla, the stroma, and then the T cell zone. But what's kind of interesting was we noticed that there is a large number of genes, and many of these genes actually overlapped across a number of different regions. So we were kind of interested in assessing which genes were differentially expressed in a manner that was spatially restricted or only associated with a single region. And when we took a look at it from that scope, what was actually quite interesting to us was we noted that the lymphoid follicle, so these regions within the PLN or the lymph nodes that are going to contain naive B cells, were associated with the highest number of differentially expressed genes in a spatially restricted manner. So that prompted us to take a little bit deeper look at what genes were differentially expressed within the lymphoid follicles within these donors. And we had a number of different hits. So you can see that we tried to focus on genes that had a great higher magnitude in log fold difference. So these are all genes that were different by at least one log fold change and are significant, noted by below. And although there's a number of different genes that we can touch on, one of the top features that popped out to us that was most upregulated in the type 1 diabetic follicles was this gene LTB, or encodes lymphotoxin beta. So we saw that this was the most significantly upregulated gene within the lymphoid follicles, kind of prompting us to take a deeper look at the expression pattern of this gene within the PLN. So we know from the literature that lymphotoxin beta is going to be predominantly expressed by cells of lymphoid origin, so thinking our T and our B cells. And when we just took a look at the spatial localization of the expression of this gene, we just mapped this across the various regions, we can see an expected pattern of expression showing that lymphotoxin beta was most enriched within the T cell zone and the B cell follicles respectively. So that goes aligned consistent with the pattern of expression that we would expect to see. When we plotted this spatially to see if there was any kind of discordance in the spatial expression pattern, we can see that this actually went ahead and tracked with what we still would have expected. We see that the greatest amount of lymphotoxin beta expression is localized to the B cell follicles, followed by the T cell zone, and then it kind of falls off from there. So we went ahead and took a closer look at the expression of lymphotoxin beta and some of these other differentially expressed genes that we identified within the lymphoid follicles and assessed their pattern of expression across individual donors. So when we took a look at lymphotoxin beta expression across individual donors, we can see that this upregulation appears pretty uniform across the type 1 donors relative to our non-diabetic and our autoantibody positive donors. In addition to some of the other genes that were detected as significantly upregulated, those are going to include these genes clusterin, follicular dendritic cell secreted protein, and the Fc epsilon receptor 2. We also noted that these genes appear to be pretty uniformly upregulated within the lymphoid follicles of these type 1 diabetic donors. So this kind of prompted us to kind of consider, what cells these transcripts could be derived from or what cells could be responding to these. And as we kind of searched through the literature, a single cell target kept continuing to pop up, and that's going to be follicular dendritic cells. And these are going to be a stromal cell population resident of the lymphoid follicles that are going to be important for both presenting antigen to naive B cells and then also for establishing kind of the chemokine gradient that's necessary for maintaining proper follicular structure. And when we took a look at the literature, this is a paper that was taking a look at the genes that were identified by single cell RNA sequencing that were identified to be highly enriched within follicular dendritic cells or specific to follicular dendritic cells. And we can see that genes that are represented among those targets are going to be clusterin and follicular dendritic cell secreted protein, making us feel a little bit more confident that these particular cell types are the ones that are potentially responding to this gene. Additionally, with respect to Fc epsilon receptor 2, this is taking a look at the expression of this particular product both at the gene and at the protein level. And we can see that follicular dendritic cells specifically are going to have the highest relative expression compared to other stromal cell populations of the lymph node. And if we compare its protein expression by flow cytometry, even respected to lymphocytes, we could see that this particular product is the most highly expressed by follicular dendritic cells. So this made us wanted to kind of take a look, a little bit more closely at, potentially is there any association between this upregulation of lymphotoxin beta and potentially the responsiveness of follicular dendritic cells in a manner that is dependent on LTB. So what was actually pretty fortunate for us is about 2 decades ago now, this group went ahead and did a characterization of downstream gene targets of lymphotoxin beta, specifically within follicular dendritic cells. So what they came down to is a list of 12 genes that were identified by either identifying genes that were highly enriched in follicular dendritic cells and genes that were most downregulated in follicular dendritic cells after blocking activation of the lymphotoxin beta receptor. And that's how they came down to this list of 12 genes. So genes that are specific to FDCs and that are stimulated specifically by lymphotoxin beta. And what you'll notice is that one of the genes that they identified was clusterin, which we were able to identify within our data set. But when we looked at the expression of these genes across our individual donors, what was actually quite interesting was we noticed a pretty strong enrichment of this lymphotoxin beta responsiveness signature, not in our type 1 diabetic donor, surprisingly, but really within our multiple autoantibody positive donors. So even though this observation is a bit discordant with the time point of when we're observing lymphotoxin beta being upregulated, it does kind of send an indication that there is some connection between this upregulation and potentially the responsiveness or the function of follicular dendritic cells within the lymphoid follicles. So I've talked a lot about lymphotoxin beta, but some of us may not be familiar with lymphotoxin beta is or what it does. So just to kind of catch everyone up with that, lymphotoxin beta is going to be a gene that's a member of the tumor necrosis factor superfamily, and the actual gene locus is going to be located within the TNF locus. That's going to be situated within the HLA class 3 locus, flanked between the class 2 and the class 1 locus. And the encoded protein is going to form a heterotrimer, 2 parts lymphotoxin beta to 1 part lymphotoxin alpha, and it's going to bind its only known receptor, which is going to be the lymphotoxin beta receptor. There is another ligand that's known for the lymphotoxin beta receptor, and we'll talk a little bit more about this in the following slides. And that's going to be encoded by another member of the TNF family of ligands, which encodes the protein called LIGHT, but we'll talk about that in the next coming slides. So what else do we know about lymphotoxin beta? What cells can respond to it? So as I mentioned, the only known receptor is going to be the lymphotoxin beta receptor. And based off of the literature and other profiling experiments experiments, we know that lymphotoxin beta receptor, both at the RNA and protein level, is going to be most highly expressed by cells of stromal lineage. So this will include cells like your fibroblasts, your endothelial cells, and those follicular dendritic cells that I just mentioned. Interestingly, when we consider the expression of lymphotoxin beta receptor in the context of immune cells, what's pretty interesting is you'll notice that it's not expressed by lymphocytes, despite them being the only cells that really express the ligand. However, the immune cells that will express the receptor are going to be predominantly these myeloid-derived antigen-presenting cells. And it's been shown that signaling through LTBR does have some impact on the development of these myeloid-derived antigen-presenting cells and also on their functionality. So there is some interesting interplay that's going on within that interaction. So what happens when the lymphotoxin beta receptor is activated? Well, that's going to go ahead and induce this downstream signaling cascade, which is going to induce the activation of the non-canonical NF-kappa B pathway. And some of the more characterized downstream products of activating the signaling cascade in these stromal cells are going to be a number of different chemokine products. And in addition to these chemokine products, also a number of different adhesion molecules and also supportive cytokines that can, have an influence on either supporting the function of certain cell types, but also can play a major impact on the recruitment of immune infiltrates into the tissue. So this is kind of an important consequence of the signaling cascade to consider. And the last thing that I wanted to mention about lymphotoxin beta is that this is going to be a contact-dependent signaling method. So for example, if we consider what we know about TNF and then also lymphotoxin alpha, it's known that this can signal through soluble TNF and lymphotoxin alpha. But what's specific to lymphotoxin beta, it's been shown that this really happens in a contact-dependent manner. So this is really where I think utilizing our spatial transcriptomic approach is going to be quite powerful, because we can actually directly see whether or not we have upregulation or co-expression of lymphotoxin beta, its receptor and downstream genes within adjacent cells that we could potentially suggest are co-localized and interacting. So I've talked a little bit about this lymphotoxin beta signature and this pathway, but one of the things that we wanted to assess is whether or not this is a pathway that is relevant in the pancreas. Number one, based off of the kind of hypothesis that lymphotoxin beta signaling within the pancreas can potentially influence the local chemokine profile, which can have an influence on the immune infiltrates and recruitment into the pancreas in a non-antigen specific manner. So to kind of follow up on some of these initial findings that we had from our first preprint, we wanted to go ahead and examine the pattern of lymphotoxin expression, both from the ligand and the receptor within the pancreas in specifically short duration type 1 diabetic donors. So within this cohort, this is a separate cohort that we're analyzing by spatial transcriptomics, however, not utilizing the Visium technology, but in this case using the Xenium technology, which is going to be an imaging-based technique. We're going in and examining the expression of these pathways within short duration pancreas. And for this, we've assembled a cohort of 22 donors. You can see that we have 8 non-diabetic donors, 2 autoantibody positive donors, and 12 type 1 diabetic donors, with a total of a little bit over 18 million representative cells across this entire cohort. In terms of the short duration, these are all going to be either onset cases or just under a year and a half of disease duration. So pretty proximal to that point of onset. And you can see these are the kind of broad level annotations of cell types that we're able to identify reliably across all of these donors. So those are going to be inclusive of not only the expected endocrine and exocrine and ductal cell types within the pancreas, but also a diverse number of immune cell types such as T, B cells, myeloid cells, and also some pretty rare cell types that are pretty underrepresented in other data sets, such as like neuroendocrine-like cells and neuronal cells. But we really wanted to focus here specifically on the expression of lymphotoxin beta receptor and its ligands in the pancreas, because that seems to be pretty undercharacterized, especially in the context of type 1 diabetes. So here we're just examining the expression of lymphotoxin beta receptor, and this is going to be visualized by this dot plot. And we can go ahead and take a look at where this gene is being expressed across these different cell subsets within the pancreas. And what was kind of surprising to me, I didn't expect it. It seems like the highest level of expression is going to be associated with the acinar cells within the pancreas, but we also see expression across endocrine cells, ductal cells, and also stromal cell populations, much like we would expect. If we examine the expression of lymphotoxin beta across disease groups, one of the things that we note, if we just kind of take a look at it kind of broadly across the entire tissue area, I don't really notice any areas of localized upregulation that would suggest that maybe one compartment or one particular region has any greater expression of lymphotoxin beta receptor across the pancreas. And if we take a look at it from that particular scope, just taking a look at the expression of this gene across the entire tissue area, we can actually see that there doesn't appear to be any difference in expression between non-diabetics, autoantibody positive donors, and our type 1s. However, if we kind of break this down and take a look at the expression across disease groups, across cell types, what was actually quite interesting is that we see that the acinar cell seems to express a greater amount of lymphotoxin beta receptor in the type 1 diabetic donors relative to the other groups. Also, kind of interestingly, it seems like there is some level of upregulation within neuroendocrine-like cells in the autoantibody positive donors as well. Although I just do want to, kind of remind everyone that we have an N of 2. So, the amount of cells that we have and the number of donors, we'd have to kind of augment this group to say a little bit more about that. But we do see that there is some difference of this particular gene, the receptor, across certain cell types based off of disease groups. So this is pretty interesting. Now, if we assess the expression of lymphotoxin beta specifically, we can see that it's actually elevated within type 1 diabetic donors. So we have 3 representative non-diabetic sections to the left, 3 representative type 1 diabetic donors to the right. And, it's pretty clear to see here that we do have a greater expression of this gene within the type 1 donors. We can see that the spatial pattern of expression can kind of vary between pretty diffuse, kind of how we see in 65, 66, or we can see some instances of localized upregulation as we see on the latter 3. But in general, the trend is that the expression of this is greater within the type 1 donors. And that's not necessarily surprising, right? We know that lymphotoxin beta is going to be expressed by lymphocytes just in general. And this upregulation is clearly going to be associated with the greater amount of lymphocytes that are present within the inflamed type 1 diabetic pancreas. But when we actually took a look at the expression of lymphotoxin beta across our T and our B cells, and we consider that across disease groups, we see that there is some amount of lymphocytes present even in the non-diabetic kind of homeostatic condition of the pancreas. And what we actually see is that we have a greater amount of lymphotoxin beta expression within our autoantibody positive and type 1 diabetic donors relative to those non-diabetic T and B cells that are found within the pancreas. So it's not just necessarily the matter of a greater frequency of lymphocytes within the pancreas, but also a matter of these lymphocytes expressing lymphotoxin beta at a higher amount relative to those non-diabetic lymphocytes. And what I wanted to present here is just one representative donor that showed this really clean and strong association between co-localization of lymphotoxin beta and chemokine expression will be found in this representative type 1 diabetic donor. So again, what I'm plotting here is the expression of lymphotoxin beta receptor in the purple. And we can see, as we saw in the other representative slides, it's a pretty diffuse expression, not really localized in any particular way. Here, we're going to be visualizing the endocrine islets based on the expression of chromogranin A expression, and you can see where we have our individual islets kind of dispersed across the pancreas. And this is not going to indicate between insulin containing or insulin depleted. This is just, all of the islets based off of chromogranin A expression. So we went ahead and took a look at the expression of the lymphotoxin beta receptor ligands, including lymphotoxin beta and TNS SF14, which encodes light, which is that other ligand for the receptor. And what we noticed was that we detected almost no expression of TNF-SF14, which encodes light. However, we did detect localized expression of lymphotoxin beta within the pancreas of this type 1 diabetic donor. So we know that this is going to be associated with lymphocytes. So we plotted the expression of canonical B and T cells, so the genes encoding CD20 and CD3. But in addition to that, we also plotted the expression of chemokines CCL19 and CCL21, which are pretty well characterized in their inducibility from lymphotoxin beta. And what we actually saw was a really strong co-localization signature between lymphotoxin beta, these chemokines, and then MS4A1, which encodes CD20 and CD3. So kind of indicating that both B and T cells expressing lymphotoxin beta are potentially contributing to this localized induction of CCL19 and CCL21. So we found this really interesting, and kind of to follow that up and to kind of wrap up this talk for today was to kind of see if there was any association between the expression of these chemokines and this localized lymphotoxin beta expression and the presence of insulin. So again, this is taking a look at just islets based off of chromogranin A, which is agnostic to whether or not insulin is actually being expressed within those islets. So we then again wanted to go ahead and look at the co-localization of lymphotoxin beta, these chemokines and CD3, which we can see visualized to the left. And then we also went ahead and followed up by staining this particular section with insulin. So this is taking a look at the immunofluorescence staining of insulin. And we can actually see is that the areas that have this co-localization signature appear to contain islets that are mostly, if not completely, insulin depleted, whereas the regions that lack this chemokine co-localization signature still contain positive insulin staining. So this suggests that the presence of this lymphotoxin beta chemokine signature is associated with the loss of insulin producing beta cells within those regions. So this is one of the things that I'm hoping to follow up within our cohort and see if we have more representative cases that kind of demonstrate this particular feature. And so that's all that I had for today. I just want to acknowledge the folks in my lab and our collaborators, both at UPenn and here at NPOD, for their help in this work. And I'd be happy to take any questions. Thank you so much for your attention.
Great talk. Would anyone like to drop a question in the chat, or feel free to raise your hand or unmute yourself?
Hey, Miguel. It's Greg.
Hey, Greg.
Great talk, man. So I have a question about the second set of data with was that probe base, the one with 18 million cells?
Yeah, that's going to be a probe-based technique.
Right. Okay. So for your probe-based data, the autoantibody positive donors, are those multiple autoantibody positive or just single?
Yeah, so one of them is single, but that single autoantibody positive donor is anti-insulin, and then the other donor is multiple, if I remember correctly, they're IA2 and zinc transporter. So even with the single GAD donors in this cohort, we're trying to bias that towards those that are non-single GAD, so more single insulin and other antigens if possible.
The higher risk. Were they young donors or not?
Yeah, so one of the autoantibody positive donors is, if I remember correctly, about 8 or 9 years old, and then the other one is approximately 17 to 18 years old. So quite a spread, but a representative young adolescent towards like late adolescent, early adult.
Okay, gotcha. Yeah, I was just curious. Like, it seems like those types of autoantibody positive individuals would be the most at risk. So like it does kind of check with the increased inflammation. Yeah, that's really interesting. And then also on the second data set, the probe-based one, could you go back to the chromogranin A? Yeah, here. Yeah. Does the chromogranin A overlap with lymphotoxin beta directly, or it seems like there's patches where there isn't chromogranin A. You can see where it's like lymphotoxin beta would sit there, like in the gaps, and also where lymphotoxin beta receptor is lacking as well.
Yeah, good question. So at least in this representative donor and the other donors that I've checked, I haven't noted a lot of strong co-localization between lymphotoxin beta, the receptor, and chromogranin. It kind of seems that where I have, higher expression of lymphotoxin beta, it seems to be in areas that are adjacent to endothelial cells. So it looks like sometimes vasculature, at least to my eyes. And sometimes I also see it in kind of regions that lack what I would think are ductal cells and really are just what seems like acinar cells. And kind of basing that off of the differential expression of the receptor in acinar cells where it appears to be elevated in the type 1 donors, it could be possible that there is some type of localized responsiveness that's occurring, not necessarily saying that the acinar cells themselves are contributing to that local chemokine expression, but it could be producing some kind of signals that can influence the phenotype or function of, maybe local fibroblasts, adipocytes that could contribute to this. So that's kind of what we've been seeing so far. And kind of that's where I'm at right now, where I'm looking at these individual sections one by one and kind of assessing the level of expression, co-localization, so on and so forth.
Yeah. Okay. And what cell type do you think is making the CCL19 and CCL21 here?
Yeah, it's another really, really good question. I would have to imagine, if I had to guess, if I had to take my best guess, some type of activated stellate cell. Not directly related to fibroblasts, but one of the things that I've seen in an unassociated paper to type 1 just kind of in vitro studies. They stimulated adipocytes with a lot of lymphotoxin beta receptor agonist, and it kind of induced some form of almost intermediate phenotype where they kind of appear to take on a little bit more of an endothelial-like phenotype in terms of their transcriptome. So I think potentially maybe excessive or aberrant lymphotoxin beta signaling within these cells to a level that's above normal homeostatic levels can potentially cause some form of dysregulation contributing to this. So that's kind of my best hypothesis at this moment.
Yeah, it's just interesting like that, lymphotoxin beta is so important for lymph node structure and development, and then you're kind of seeing like signatures of what looks like a semi lymph node type of setup in the tissue. So
And
it's just interesting.
not seeing that cellular organization just yet, or some
Right,
of
right,
those
right.
other genes that would be indicative. But yeah, it's an interesting similarity between the two.
Yeah, just a correlation at this point, but it is interesting. That's cool.
Yeah.
Thank you.
Thank you.
Wait, are you guys saying that it's almost like the immune system setting up like a lymph node field camp?
No, 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 I'm not going that
Wait.
far. I'm just
Yeah,
saying
Okay.
neither am I.
I'm not going
Wait,
that
wait.
far. There's
Well,
not enough evidence here, but I'm
well,
just
I'm going to go that far because that's what it looks like. And it kind of reminds me, Miguel, almost of like ectopic follicles that one might see in like gut mucosa. And
Huh.
so have you ever looked at that just to see if you get similar signatures, perhaps?
The gut mucosa, no, I haven't. So that could definitely be something that could be looked at in, say, like public data sets to kind of get a better sense of what potentially may be going on here for sure. I do know, for example, with other autoimmunities that have a tendency, especially in humans, to form these kind of ectopic germinal center structures, like, for example, in rheumatoid arthritis. There has been a little bit more work that's been done in that space and has shown that lymphotoxin beta signaling in kind of animal models in vitro setting does contribute directly to the formation of these types of structures. So a little bit more needs to be done to understand if such a similar mechanism is occurring in the human pancreas and T1D. But at least in other autoimmunities, there does appear to be some link that it could be directly contributing to the formation of these types of organized structures that could be, permissive to immune-immune interactions locally.
Do you see any enhanced sort of formation of lymphatic vessels in those areas? Sort of like some
That's like,
drainage could like become better or something?
yeah, no, that's a good question. I haven't looked super directly at that, but one of the things that I have noted, for example, is that at least in endothelial cell populations, it appears that they're expressing a higher amount of Lymph1 in the pancreas. So that could be indicative of more of a lymphatic vessel kind of structure that could be, again, permissive to this. So I need to take a look if that's coming directly from the vessels or endothelial cells themselves, or if it's being derived from, say, Lymph1 positive macrophages that also are detectable within the pancreas in our data set. So I'm not too sure about the source, but we are detecting that, which I would say is indicative of that potentially.
Very nice. Thank you.
Thank you.
Yeah, well, it's a real treat to have two of the authors on this call. Both Miguel and Greg are on the preprint, so thank you both for contributing. It's great discussion. I had a quick question. You noted like the highest expression of LTBR is in the Acinar cells and T1D donors. Is there any way to like do some time lapse, evaluation going back in time to sort of see how that might kick off? I mean, Acinar cells, first of all, is kind of, you were surprised by that, right?
Yeah, that's a good question. Just kind of thinking in the context of like the data that we have, right? This is all really cross-sectional and we only have so many, pseudo time points, I guess that you could say, right? But, with the data that we have, at least from the data that we collected in the preprint, we have a pretty nice range of disease duration within the type 1 pancreas. So we do have a representative like onset case, but we also have those that are longer disease duration. And within this individual cohort with the xenium that we have, this is more reflective of shorter durations. So it could be possible for us to maybe just kind of take a look independently what the expression levels look at various disease duration points and see if that could be correlated in any kind of way. Otherwise, I don't know how we could look at it in a time associated kind of way. That'd be kind of a difficulty, but one that's I think worth looking at if we could.
You just wonder if there's any other samples that have already been processed that are sitting in some repository somewhere
Sure.
also. Okay, anyone else?
actually I had another question about autoantibodies. This is just going kind of speculation mode, but like when I'm looking at lymph nodes, for example, I don't really see evidence of like germinal center expansion or activity increasing. This is more through like the TFH side of things. And maybe it's because I'm missing like the critical window of peak inflammation, et cetera. But like, do you think that maybe autoantibody advancement or antibody editing could occur in these type of structures? Like, do you see any evidence of that, or would you care to speculate on that?
Mm.
Yeah, I don't think I have any evidence to suggest one way or the other.
Okay.
However, one of the things that has been kind of interesting is, for example, one of the things that this lymphotoxin signaling can do in both stromal and myeloid-derived antigen-presenting cells is it can impact their ability to efficiently present antigen. And if we think about myeloid-derived cells, that'll be in the context of, MHC. But if we also think about stromal-derived like FDCs, is going to be in the context of immune complexes, so FC and complement receptor expression. And in addition to that, it could also affect their ability to secrete other cytokines that could be important for licensing or also support survival factors for like B cells. So one of the things that, I'm thinking is that because I haven't seen anything in my data that would be suggestive of, there being more secondary lymphoid follicles or more primary follicles or anything like that. But potentially what I could potentially speculate is on through excessive signaling through lymphotoxin beta, you could potentially make these antigen presenting cells more efficient stimulators of lymphocytes, either through more antigen presentation in their respective context or through increased expression of co-stimulatory or cytokine factors, which could contribute to that break in peripheral tolerance that we see. So I think it's kind of a matter of stimulating these APCs to a degree that makes them more efficient activators of immune cells, and that also will include the autoreactive cells that typically wouldn't receive enough stimulation to become activated under normal conditions. And that's kind of my speculation at this time.
Yeah, and also I think and B cells can also be APCs and as well, like just to add to that. But yeah,
Mhm.
no, it's very interesting. Thank you.
Thank you.
Yeah, this like a really series of great thought experiments. I just wanted to just sort of call out, last week we had Clive Wasserfall at the Sugar Science here at the think tank. He was sort of highlighting some of his work, and then he just put out the paper July 24th in Diabetes, the loss of insulin positive cell clusters precedes the decrease in islet frequency and B cell area in type 1 diabetes. And then, of course, the disappearance of the endocrine objects that Sarah Richardson sort of discovered and brought to light. How do those two pieces of the puzzle intersect with yours, if you could speculate?
That's a tough question. That's a tough
I
question on the
know.
spot, right?
It
It's
is a
almost
really tough question.
it's definitely one of those like chicken versus egg things, right? Like what is preceding
Yeah,
the loss of beta
exactly.
cells? And I'd have to imagine potentially it's kind of a concurrent thing. So I'd imagine that, beta cell death is initiated by something, in my opinion, right? Something that's maybe not directly autoimmune associated due to some sort of viral infection, some form of metabolic stress that, is going to lead to that initial immune activating an event. And I think due to genetic risk modifiers, genetic risk variants, that'll lead towards, kind of breaks in certain pathways that would normally be regulated. And that will include, for example, like the expression of lymphotoxin in response to these downstream pathways. So they kind of happen in parallel to one another,
Yeah,
in my opinion. So as death occurs, you kind of kick this kind of pathway off and it further augments itself as inflammation increases and as beta cell destruction also increases.
yeah. Some inherent fragility and then the response. A bad ballet. Okay, thank you, Miguel, and thanks, Greg, too, for jumping on. Great talk, great preprint, and we really are looking to see what comes next out of your project. Congratulations on your PhD, and we'll look forward to speaking with you again.
I appreciate it. Thank you so much for the invitation and thank you for everyone's time and attention. Thank you guys.
Thanks. Have a great rest of your day. Bye-bye.
Thank you. Bye-bye.